Bismillahirrahmanirrahim. The topic of today's lecture is immune system. Objectives. By the end of this lecture, you will be able to enlist the three lines of defense and outline their properties. Describe the types of immunity. Describe the role of white blood cells that participate in adaptive and innate immunity, and compare the active and passive immunity. Now, immunity is divided into two main classes. One is innate immunity, which is non-specific immunity, and the other is acquired immunity, which is specific immunity. Acquired immunity is again divided into two main classes. These are the humoral immunity and the other is cellular immunity. Humoral immunity is antibody mediated, while cellular immunity is T cell mediated. Now the innate immunity. Innate immunity is the inborn capacity of the body to resist pathogens. By chance, if the organism enters the body, innate immunity eliminates them before the development of any disease. It is otherwise called the natural or non-specific immunity. Innate immunity includes the following: phagocytosis of bacteria and other invaders by white blood cells and cells of the tissue macrophage system. Destruction of swallowed organisms by the acid secretions of the stomach and the digestive enzymes. Resistance of the skin and the mucous membrane to invasion by organisms. Presence in the blood of certain chemical compounds that attach to the foreign organisms or toxins and destroy them. Some of these compounds are lysozyme, which is a mucolytic polysaccharide that attack bacteria and causes them to dissolute. Basic polypeptides, which inactivate certain types of gram-positive bacteria. The complement complex, a system of about 20 proteins that can be activated in various ways to destroy bacteria. And natural killer cells, which are the lymphocytes that can recognize and destroy foreign cells, tumor cells, and even some more infected cells. Acquired immunity. Acquired immunity or specific immunity. Acquired immunity is the resistance developed in the body against any specific foreign body like bacteria, viruses, toxins, vaccines, or transplanted tissues. So this type of immunity is also known as specific immunity. It is the most powerful immune mechanism that protects the body from the invading organisms or toxic substances. Lymphocytes are responsible for the acquired immunity. There are two types of acquired immunity: cellular immunity and humoral immunity. T lymphocytes are responsible for the development of cellular immunity, and B lymphocytes are responsible for humoral immunity. Adaptive immune system or acquired immunity has two arms: humoral immunity and cell-mediated immunity. Humoral immunity is provided by B lymphocytes, while cellular mediated immunity is provided by T lymphocytes. Humoral immunity can recognize protein, polysaccharides, phospholipids, and nucleic acid antigens, while cellular immunity can recognize only protein antigens. Humoral immunity can act against soluble or free antigens, while cellular immunity recognizes antigens presented by antigen presenting cells with class 1 or class 2 MHC molecules. Humoral immunity provides immunity to extracellular bacteria, viruses, and toxins, while cellular immunity provides immunity to intracellular bacteria, viruses, fungi, and protozoa. Humoral immunity causes type 1, 2, and 3 hypersensitivity reactions, while cellular immunity causes type 4 hypersensitivity and causes acute graft rejection. The immune system can be divided into three basic lines of defense against pathogenic infection. The first line of defense against infection are the surface barriers that prevent the entry of pathogens into the body. The second line of defense are the non-specific phagocytes and other internal mechanisms that comprise innate immunity. The third line of defense are the specific lymphocytes that produce antibodies as a part of the adaptive immune response. This slide shows the flow chart for immunity. This flow chart shows that immunity has basically two main types: innate and adaptive immunity. Innate immunity has the components like barriers, which include skin, acidic gastric juice, microflora, epithelial cells, and complement proteins. These components are always on, means that they are always ready to fight with microorganisms, and is considered to be the first line of defense mechanism. Innate immunity also contains neutrophils, which are phagocytic cells, and antigen presenting cells like macrophages and dendritic cells. These cells give immediate response when a pathogen comes in contact with these. So they are considered to be the second line of defense mechanism. While adaptive immunity is activated when a specific pathogen enters into the body, and it gives strong and delayed response. So it is considered as a third line of defense mechanism. Now the first line of defense. The primary defense against infectious diseases are the surface barriers that prevent pathogens from entering the body. These surface barriers include intact skin and mucous membranes. Both the skin and the mucous membranes release chemical secretions which restrict the growth of the microbes on their surfaces. If pathogens cannot enter the host body, they cannot disrupt normal physiological functions and cause disease. Now the second line of defense. The second line of defense against infection are the non-specific cellular and molecular responses of the innate immune system. These defenses do not differentiate between different types of pathogens and respond the same way upon every infection. Phagocytic leukocytes migrate to the infection site and engulf foreign bodies. Inflammatory responses increase capillary permeability at the infected sites, recruiting leukocytes but leading to localized swelling. Antimicrobial proteins such as cytokines and complement proteins regulate immune activity within the body. Fever increases body temperatures to activate heat shock proteins and suppress microbial growth and propagation. Now coming to the third line of defense. The final line of defense against infection are the lymphocytes that produce antibodies to specific antigenic fragments. Each B lymphocyte produces a specific antibody, and the body has millions of different B lymphocytes capable of detecting distinct antigens. Helper T cells regulate B lymphocytes cell activation. ensuring that antibodies are only mass produced at the appropriate time. Both B lymphocytes and T lymphocyte cells will differentiate to form memory cells after activation, conferring long-term immunity to a particular pathogen. Now, the role of white blood cells in immunity. White blood cells play an important role in defense mechanism. These cells protect the body from invading organisms or foreign bodies either by destroying or inactivating them. However, in defense mechanism, each type of WBCs acts in a different way. Now the neutrophils. Neutrophils are most abundant and most motile WBCs present in our body. Neutrophils play an important role in the defense mechanism of the body. The neutrophils are the free cells in the body and wander freely through the tissues, and practically no part of the body is spared by these leukocytes. Neutrophils are released in large number at the site of infection from the blood. All the neutrophils move by diapedesis towards the site of infection due to chemotaxis. Neutrophils surround the area and get adhered to the infected tissues. Now the neutrophils start destroying the invaders. First, these cells engulf the bacteria and then destroy them by means of phagocytosis. Now eosinophils, which is another type of WBCs, eosinophils play an important role in the defense mechanism of the body against the parasites. Its count increases also during allergic diseases like asthma. Eosinophils are responsible for detoxification, disintegration, and the removal of foreign proteins. Eosinophils are neither markedly motile nor phagocytic like neutrophils. Some of the parasites are larger in size. Still, eosinophils attack them by some special type of cytotoxic substances present in their granules. Basophils. Basophils play an important role in healing processes. Basophils also play an important role in allergic or acute hypersensitivity reactions. This is because of the presence of receptors for IgE in basophil membrane. Functions of basophils are executed by the release of some important substances from their granules, such as heparin. Heparin is essential to prevent the intravascular blood clotting. Histamine, slow-reacting substances of anaphylaxis, bradykinin, and serotonin. Proteases and myeloperoxidases. These enzymes destroy the microorganisms. Cytokine like interleukin 4 accelerate inflammatory responses and kill the invading organisms. Mast cells. Mast cell plays an important role in producing the hypersensitivity reactions like allergy and anaphylaxis. When activated, the mast cell immediately releases various chemical mediators from its granules into the interstitium. Two types of substances are secreted by the mast cell. Preformed mediators. These substances are already formed and stored in the secretory granules. These substances are histamine, heparin, serotonin, hydrolytic enzymes, proteoglycans, and chondroitin sulfates. Other type is newly generated mediators. These substances are absent in the mast cell during resting conditions and are produced only during activation. These substances are arachidonic acid, derivatives such as leukotrienes C, prostaglandin, and cytokines. Monocytes. Monocytes are the largest cells among the leukocytes. Like neutrophils, monocytes are also motile and phagocytic in nature. These cells wander freely through all the tissues of the body. Monocytes secretes interleukin 1, colony stimulating factor, platelet activating factor. Monocytes are the precursors of the tissue macrophages. Now the big eaters, the macrophages. Macrophage is a large phagocytic cell derived from monocyte. Matured monocytes stay in the blood only for few hours. Afterwards, these cells enter the tissues from the blood and become tissue macrophages. Examples of tissue macrophages are Kupffer cells in the liver, alveolar macrophages in the lungs, and macrophages in the spleen. Macrophages perform two main functions: phagocytosis of the pathogens and antigen presenting cells. Lymphocytes. Lymphocytes play an important role in immunity. T lymphocytes and B lymphocytes are the cells of acquired immunity. Types. T lymphocytes. T lymphocytes are responsible for the development of cellular immunity. This type of acquired immunity is achieved through the formation of large numbers of activated T lymphocytes that are specifically crafted in the lymph nodes to destroy the foreign agents. This type of immunity is called cell-mediated immunity or T-cell immunity because the activated lymphocytes are T lymphocytes. Now B lymphocytes. B lymphocytes are responsible for the development of humoral immunity. The body develops circulating antibodies that are capable of attacking the invading agent. B lymphocytes produce the antibodies and natural killer cells. This slide shows another classification of acquired immunity. Acquired immunity is divided into active immunity and passive immunity. In active immunity, the individual's own immune system is the cause of the immunity, while in passive immunity, immunity is transferred from one person or an animal. Active immunity is again divided into natural and artificial immunity, while passive immunity is again divided into natural and artificial immunity. In natural active immunity, antigens are introduced through natural exposure, while in active artificial immunity, antigens are deliberately introduced in a vaccine. In natural passive immunity, antibodies from the mother are transferred to her child across the placenta or in a milk, while in artificial passive immunity, antibodies produced by another person or an animal are injected. Now, actually, what is active immunity? Active immunity is the production of immunity against particular organisms after exposure. It is of two types: natural active immunity and artificial active immunity. Natural active immunity, this immunity develops by natural processes like infections. For example, the infection like smallpox are cured by the active function of the immune system. And artificial active immunity, here instead of natural infection, infection is created artificially by using various types of vaccines, for example, polio vaccine, cholera vaccine, etc. Passive immunity. Acquiring of the antibodies passively from an immunized donor to a non-immunized recipient is known as passive immunity. Here it is again of two types: natural passive immunity and artificial passive immunity. Natural passive immunity, it occurs when antibodies are transferred from one donor to the recipient in a natural manner. For example, transfer of antibodies from the mother to the fetus through the placenta, like in immunoglobulin G. Artificial passive immunity, the transfer of antibodies and sensitized lymphocytes from the immunized donor to the non-immunized recipient artificially. For example, antibodies produced in the horse serum and genetically modified organisms. This brings us to the end of this lecture. Hope you understood the lecture. Thank you.